you will read several rules that we hope all participants will pay attention to. Number 1, all participants are encouraged to turn on their cameras. Number 2, please rename your Zoom display name to the following format: institution _ full name. Number 3, during this event, please mute your microphone unless you have been given permission to speak by the moderator. Number 4, participants are encouraged to use the virtual background that has been provided in the chat box. Number 5, during this event, we will be using our website, www.fkunram.ac.id, to conduct the pre-test, post-test, and participants' feedback. During the question and answer session, participants are encouraged to actively participate. Questions may be asked in both English and Indonesian. After this event ends, you will send in a participant feedback link through the chat box. We hope all participants will fill in the form. And we would also like to remind all participants to fill in the attendance list that we have sent through the chat box. Thank you. Ladies and gentlemen, please allow me to once again read the rules for this event. Number 1, all participants are encouraged to turn on their cameras. Number 2, please rename your Zoom display name to the following format: institution _ full name. Number 3, during this event, please mute your microphone unless you have been given permission to speak by the moderator. Number 4, participants are encouraged to use the virtual background that has been provided in the chat box. Number 5, during this event, we will be using our website, www.fk.unram.ac.id, to conduct the pre-test, post-test, and participants' feedback. During the question and answer session, participants are encouraged to actively participate. Questions may be asked in both English and Indonesian. After this event ends, we will send in a participant feedback link through the chat box. We hope all participants will fill in the form. And we would also like to remind all participants to fill in the attendance list that we have sent through the chat box. Thank you. Ladies and gentlemen, during this event, we will be using our website www.fk.unram.ac.id to conduct all pre-tests, post-tests, and participants' feedback. Thank you. The Honorable Vice Dean for Student Affairs, Faculty of Medicine, University of Mataram, guest lecture speaker, invited guests, students, and all participants of the guest lecture titled One Health Approach to Tackling Clostridium difficile Infection. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi. Good morning, ladies and gentlemen. My name is Rukwanggana Satya Pratiwi, and I am the Master of Ceremony in this guest lecture. In the developing world, collaboration of various sectors through the One Health approach holds a significant role in addressing and preventing zoonotic diseases to achieve better public health outcomes. Today, we're gathering here to know more about how we are going to implement the One Health approach to tackle Clostridioides infection specifically. First of all, I would like to invite the Vice Dean for Student Affairs, Faculty of Medicine, University of Mataram, to deliver opening remarks and officially open today's guest lecture. To Dr. Rohadi, Consultant in Neurosurgery, FHS, FENPS, time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. I'm Rohadi from Medical Faculty of Mataram University. Good morning, ladies and gentlemen. I would like to extend my warm welcome to our esteemed speaker, Mrs. Su Chan Lim, PhD, and to our moderator, Mrs. Dewi Suryani, MD. And welcome to all distinguished participants, researchers, and students. It's my pleasure to be here to open our guest lecture today, One Health Approach to Tackling Clostridium Infection. The number of cases of Clostridium difficile infection has increased in past decade. In Indonesia itself, about 150,000 people are infected annually. The spread of the infection is especially common in hospital setting. Hospitals are an environment where humans interact, and the spread of antibiotic-resistant germs within the setting of some of concern in One Health initiative. I'm looking forward to hear more from our esteemed speaker regarding this collaborative approach and how it can contribute in our effort to tackle the infection. Before concluding, I would like to convey my appreciation to our esteemed speaker and moderator for their time and support in contribute to this lecture. I thank you all participants most warmly from your participation and wish you all success. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Dr. Rohadi, Consultant in Neurosurgery, FHS, FENPS. Before we start our guest lecture, there is a pretest which will be conducted for 5 minutes. The pretest link has been sent in the chat box, and participants will be redirected to our website where the pretest will be done. We wish all participants to fill in this pretest. Thank you. To all participants, we have sent the pretest link in the chat box, and participants will be redirected to our website where the pretest will be done. We wish all participants to fill in this pretest. Thank you. Time is up. Thank you to all participants for completing the pretest. Ladies and gentlemen, now we will proceed to the main event, our guest lecture. The session will be led by the moderator, Dr. Dewi Suryani, Master of Infectious Diseases. Dr. Dewi, graduated from the Faculty of Medicine, Brawijaya University, and continued her postgraduate study with a focus of infectious diseases at the University of Western Australia. Dr. Dewi has been working as a lecturer in the Faculty of Medicine, University of Mataram for 16 years. An extensive range of research, including malaria, dengue, tuberculosis, COVID-19, and many more had been carried out by Dr. Dewi. To Dr. Dewi Suryani, Master of Infectious Diseases, time is yours.
Hi, hello everyone. Good morning. Assalamualaikum warahmatullahi wabarakatuh. I am extremely honored to be the moderator of this guest lecture. This is our first guest lecture in 2022, and it is supported by the Partnership Unit and also Avicenna from the Faculty of Medicine, University of Mataram. I am delighted to see all the participants here eager to learn on the concepts of One Health. One Health is a concept that emphasizes on the need for collaboration between different sectors, which include human health, animal health, and also the environment in tackling different aspects of infectious diseases. That is why I am pleased to see participants from different study backgrounds come together in this event and understand more about this concept. This is a subject that we know that is taught in the first year of the medical curricula. And I hope the presentation that would later be presented by Dr. Su-Chan Lim would inspire students to understand more on the importance of collaboration between different expertise and also motivate and inspire students to conduct research in this field. As we know together that in regards to Clostridium difficile, it is also something that is closely related with the condition here in Lombok and also in Western Nusa Tenggara, as there are many cattle farms in this area. This talk would be a great example on bridging the concept to practice One Health in regards to One Health through its implementation in research. So with us today here we have Dr. Su-Chan Lim, a dear friend of mine at the University of Western Australia. Hello, Su-Chan. We are very delighted to have you here.
I'm very happy to be here.
Yep. And currently she's a staff member at the School of Biomedical Science at UWA and also under the Riley Laboratory. So Su-Chan and I first met during our postgraduate study back in 2009. And since 2011, she's been consistent in conducting research in Clostridium difficile. So here is a brief CV of her, as you can see. So Ms. Su-Chan Lim conducted her, next slide, please. Yep. So she conducted her bachelor degree at UWA and then continued for her master's degree with the same field as what I did before, and that is in Master's Infectious Disease at UWA. Specifically, this was the starting point, I guess, where she was passionate in regards to Clostridium difficile. And so since then, she's been focusing her research and focus area on this aspect. And then in 2019, she completed her PhD degree at UWA and also in the focus area of Clostridium difficile. Next slide. Okay, so she has, there's many appointments that she has previously experienced. So as staff also at UWA and also at Edith Cowan University. Next slide. And her current research projects are in reducing hospital-identified Clostridium difficile infection by reducing environmental sources of C. difficile, and also C. difficile in children with cancer, and also in regards to development of vaccines in swine or pigs. Next slide. Okay, so this is the extensive work that she has done. It's quite an incredible list in the field of Clostridium difficile. So we're really happy that you're here to share all your research experience and So the students here could be inspired with the work on Clostridium difficile and also on One Health. Next slide. Okay, so this is also the list of her work in C. difficile. Next slide. Okay. So without further ado, we would start the presentation by Dr. Su-Chan Lim and followed by a Q&A session. And as stated before by the MC, we encourage students to later ask questions through chat box or ask directly in the Q&A session. You could either ask the question in English or in Indonesia, and we'll do our best to translate the questions from Indonesia to English. So don't worry about the communication barrier, because by all means, we would do our best to translate it for you. So without further ado, let's welcome Dr. Su-Chan Lim, and we'll dive into her talk entitled of One Health Approach to Tackling Clostridium difficile infection. So would the host of this session please share the screen to Ms. Su-Chan Lim? Thank you.
Okay, so do I start the screen sharing now?
Yeah.
Okay, so I already recorded everything. So right now I'll play it for you. So let's do this, share, and video. Can you guys see the slides? Play.
Hi everyone, I'm Su-Chan. I'm a postdoc from the University of Western Australia. Thank you so much for inviting me to do this talk. And today I'll be talking to you about Clostridium difficile and One Health. And before I forget, here are my email address and Twitter handles. So if you have any questions, feel free to let me know. Thank you. Today I'll be talking to you about studying humans, animals, environments, as well as the One Health approach that we can use to solve these One Health issues, followed by some of the current studies that our research group is undertaking. So let's start with C. diff in humans. So what is Clostridium difficile? Clostridium difficile is a spore-forming pathogen. It can persist in the environment for a very long period of time. It's also resistant to extreme temperature, desiccation, UV lights, and common disinfectants, making it very hardy and hard to kill. Clostridium difficile infections is a toxin-mediated disease caused by 3 different types of toxins: toxin A, toxin B, and binary toxins. The symptoms can include diarrhea, and in serious cases, pseudomembranous colitis, toxic metacolons, and death. The risk factors include exposure to C. diff spores, as well as a history of antibiotic usage and old age over the age of 65. Now, let's talk a little bit about the history of Clostridium difficile. C. diff was first isolated in humans in 1935 from the meconium and feces of healthy infants. Now, because infant doesn't have the receptors for the toxins produced by C. difficile, it doesn't cause any diseases in the babies. But in 1950s, that was the golden era of antibiotic discovery. And what happens after we discover antibiotics? We immediately started using it, especially the broad spectrum antibiotics. And many of those have activities against the gut microbiomes. And the gut microbiomes will die, but then C. difficile will come in and have a look around, and it realizes that these adult guts now look like a neonatal guts because there's no competition around. So C. diff will come in, set up shop, produce toxins, but in this case, they're producing toxins in adults that has consumed antibiotics, and because there's no competition around, C. diff will flourish. And in 1960s, we are seeing an increased number of pseudomembranous colitis cases. Nobody knows why or what causes the increase. And in 1970s, we've isolated C. difficile from hospitalized patients who were undergoing antimicrobial therapy. But then nobody made the connection that C. difficile actually causes the pseudomembranous colitis until 1978. In a span of around 2 to 3 months, around 3 papers were published in the same year. They all claim to found the same thing, which is C. difficile causes pseudomembranous colitis, and it's all because of antimicrobial usage. And because C. difficile is linked to antimicrobial usage, it eventually became a hospital-associated infections, quite well-known hospital-associated infections, because there's a lot of people in the hospital that are using antibiotics and then they're old and have comorbidity as well. So this is Sir Charles Gunner Hospital in Western Australia, and my lab and office is based here around this area, and sometimes we call this hospital Charlie's Hospital. So when I'm saying Charlie's, I'm talking about this hospital. I'm not talking about a person named Charlie, you know. So just now we finished the last slides talking about linking C. difficile to causing pseudomembranous colitis in 1978, and after that it became a hospital-associated infections. So now we're going to have a look at what happened at Charlie's in between 1983 to 1992. So what happened was there was an increased number of CDI cases, as you can see from this chart over here, and majority of the cases were elderly and female. And this paper over here was published in 1994. I was probably in primary school back then, and this is my supervisor, Professor Tom Riley. He was also my supervisor for my PhD, which I finished in 2019. So in that year, the hospital decided to have a look at what's going on, and they did a little bit of investigations. And what they found was it was because of increased use of the generation cephalosporins. As you can see, increased use of cephalosporins, increased number of CDI. And that's because C. difficile is intrinsically resistant to these drugs. And then in the following years, the hospital decided to use less third generation cephalosporins. And as you can see from this graph over here, the use of antibiotic has gone down quite significantly. And that led to a decrease in CDI cases. So while everyone was focusing on what was happening in the hospital, something else was happening in the animal sectors. So in 1990, cephalosporin, which is also a third generation cephalosporin, was licensed to use in animals in North America. So that happened in the 1990s. And around 10 years later, we were seeing an increased number of CDI cases in humans in the US. The cases just went straight up, as you can see from this graph over here. And that was because of a newly emerged rabbit type called rabbit type 027. So rabbit type 027 is hypervirulent. It causes more severe diseases, and it produces all three toxins: toxin A, toxin B, and binary toxins. It's also resistant to fluoroquinolones. It acquired the resistant genes twice independently of each other. And because everyone was focusing on what's happening in a hospital, and also because C. difficile infection is a well-known hospital-associated infections, nobody was paying attention to what was happening in the community. Community-associated cases were completely missed in North America. And in Australia, we also decided to license cephalosporin for animal use in 2000. And guess what? In around 10 years later, in 2010s, we were also seeing an increased number of CDI cases. Now let's talk a little bit about the emergence and spread of rabbit type 027. Canada was the first country to report such a significant increase in CDI cases that was linked to rabbit type 027. In between 1991 to 2003, the case number increased by 4 times. And among the elderly population, it increased by more than 8 times. And both the complication and mortality rates more than doubled. And that was because of 2 different lineages of rabbit type 027 that are resistant to fluoroquinolones. And if you have a look at this map over here, it shows that the 2 lineages basically arrived somewhere in North America, and then they were traveling back and forth between North America and Europe and UK quite a few times before they spread to other parts of the world. So how important is C. difficile? Well, it's ranked as one of the top 5 urgent AMR threats by the CDC in the US.
Sorry to bother you.
100,000 infections and
Hello,
29,000
Suchan.
deaths per year in the US, and that's roughly around 6% mortality rate.
Could you hear me?
Australia, they equate to roughly around 11 deaths per week. It causes the healthcare system a lot of money, mostly because of increased hospital stay and treatment costs. Now, community-associated CDI cases is actually on the rise.
Can
In the
you
US,
hear my
40%
voice?
of CDI cases are community-associated, and in Europe, that's 15%. In Australia, 30% of the cases are from the community.
Hello, Suchan. Can you hear my voice?
By David Ayer, who's in the
Yes.
Yep. We're hearing the audio is too low at our
Okay.
receiving point. Would it be possible to make the audio clear? Have you shared the audio? Or is it on your screen? Sorry, I haven't turned on my video.
It's on my speaker.
Okay.
Yeah.
And have you shared it through the share screen, the audio?
Share screen.
Yeah, when you put share screen.
Oh, share sound. Now I get it.
Yeah.
Yep. Got it.
Yep.
Thank you. Sorry. Okay,
No.
let's continue.
Yep, we'll see how that goes. You can just continue from where you.
Okay. It's 900 plus isolates from human CDI cases and perform whole genome sequencing followed by stem analysis. And his study shows that 45% of those CDI cases were actually genetically distinct from all previous cases. It shows that there's quite a significant proportions of the CDI cases over there are not acquired from another symptomatic patients, which totally refute the traditional notions that CDI is a hospital-associated infections acquired from another case. And this study shows that there must be another source causing all these CD infections outside of the hospital environment. Now let's look at what ribotype is circulating around Australia. The top ribotype in Australia is always ribotype 014/020. That's this one over here, and in red, and can be found in all states. And then the second most common ribotype is 002, followed by 056. Now that one is actually quite consistent. They're always on the top 5. Sometimes there are new strain that will pop in every now and then. For example, ribotype 012. That one is quite new. We think it might be originated from Asia. And then ribotype 017 is also very prevalent in Asia as well. So that may be where it's coming in from. So now let's have a look at what's going on in Asia. So this is a study performed by Deirdre Collins. She's a postdoc in our research groups, and this study was funded by Otsuka, which is a pharmaceutical company based in Japan. So they wanted to make vaccines for C. difficile, and before they do that, they wanted to know what strain is circulating around Asia, which is why they commissioned us to perform this study. So in Asia, the most common ribotype is actually ribotype 017. But it's ribotype 018 that's the most common in South Korea and Japan. So this study also included some strain from Indonesia, but we only have 7 from Indonesia. So if you guys want to give us some sample, feel free to contact me.
Okay. Suchan, so sorry to interrupt again.
Yes.
It turns out that many of the participants couldn't hear the several talk. The talk in the first.
I can start from the beginning.
Oh yeah, would that be okay with you?
Yeah, that's completely fine.
Okay, thank you so much, Suchan.
Okay, you guys just have to focus more on my spend more time on my presentation. Then that's fine.
Yep. Okay. Thank you.
Okay. Starting from the beginning, everybody.
Okay. Thank you, Suchan.
Hi everyone, I'm Suchan. I'm a postdoc from the University of Western Australia. Thank you so much for inviting me to give this talk. And today I'll be talking to you about Clostridium difficile and One Health. And before I forget, here are my email address and Twitter handles. So if you have any questions, feel free to let me know. Thank you. Today I'll be talking to you about C. diff in humans, animals, environments, as well as the One Health approach that we can use to solve these One Health issues, followed by some of the current studies that our research group is undertaking. So let's start with C. diff in humans. So what is Clostridium difficile? Clostridium difficile is a spore-forming pathogen. It can persist in the environment for a very long period of times. It's also resistant to extreme temperature, desiccation, UV lights, and common disinfectants, making it very hardy and hard to kill. Clostridium difficile infections is a toxin-mediated disease caused by 3 different types of toxins: toxin A, toxin B, and binary toxins. The symptoms can include diarrhea, and in serious cases, pseudomembranous colitis, toxic megacolons, and death. The risk factors include exposure to C. difficile spores, as well as a history of antibiotic usage and old age over the age of 65. Now let's talk a little bit about the history of Clostridium difficile. C. diff was first isolated in humans in 1935 from the meconium and feces of healthy infants. Now, because infant doesn't have the receptors for the toxins produced by C. difficile, it doesn't cause any diseases in the babies. But in 1950s, that was the golden era of antibiotic discovery, and what happens after we discover antibiotics? We immediately started using it, especially the broad-spectrum antibiotics, and many of those have activities against the gut microbiomes, and the gut microbiomes will die, but then C. difficile will come in and have a look around, and it realized that these adult guts now look like a neonatal guts because there's no competition around. So C. diff will come in, start to set up shop, produce toxins, but in this case, they're producing toxins in adults that has consumed antibiotics, and because there's no competition around, C. diff will flourish. And in 1960s, we are seeing an increased number of pseudomembranous colitis cases. Nobody knows why or what causes the increase. And in 1970s, we've isolated C. difficile from hospitalized patients who were undergoing antimicrobial therapy. But then nobody made the connection that C. difficile actually causes the pseudomembranous colitis until 1978. In a span of around 2 to 3 months, around 3 papers were published in the same year. They all claim to found the same thing, which is C. difficile causes pseudomembranous colitis, and it's all because of antimicrobial usage. And because C. difficile is linked to antimicrobial usage, it eventually became a hospital-associated infections, quite well-known hospital-associated infections, because there's a lot of people in the hospital that are using antibiotics, and then they're old and have comorbidity as well. So this is Sir Charles Gunner Hospital in Western Australia, and my lab and office is based here around this area. And sometimes we call this hospital Charlie's Hospital. So when I'm saying Charlie's, I'm talking about this hospital. I'm not talking about a person named Charlie, you know. So just now we finished the last slides talking about linking C. difficile to causing pseudomembranous colitis in 1978. And after that, it became a hospital-associated infections. So now we're going to have a look at what happened at Charlie's in between 1983 to 1992. So what happened was there was an increased number of CDI cases, as you can see from this chart over here. And majority of the cases were elderly and female. And this paper over here was published in 1994. I was probably in primary school back then, and this is my supervisor, Professor Tom Riley. He was also my supervisor for my PhD, which I finished in 2019. So in that year, the hospital decided to have a look at what's going on, and they did a little bit of investigations. And what they found was it was because of increased use of third generation cephalosporins. As you can see, increased use of cephalosporins, increased number of CDI. And that's because C. difficile is intrinsically resistant to these drugs. And then in the following years, the hospital decided to use less third generation cephalosporins, and as you can see from this graph over here, the use of antibiotic has gone down quite significantly, and that led to a decrease in CDI cases. So while everyone was focusing on what was happening in the hospital, something else was happening in the animal sectors. So in 1990, cephalospore, which is also a third generation cephalosporins, was licensed to use in animals in North America. So that happened in 1990s. And around 10 years later, we were seeing an increased number of CDI cases in humans in the US. The cases just went straight up, as you can see from this graph over here. And that was because of a newly emerged rabbit type called rabbit type 027. So rabbit type 027 is hypervirulent. It causes more severe diseases and it produces all three toxins, toxin A, toxin B, and binary toxins. It's also resistant to fluoroquinolones. It acquired the resistant genes twice independently of each other. And because everyone was focusing on what's happening in the hospital, and also because C. difficile infection is a well-known hospital-associated infections, nobody was paying attention to what was happening in the community. Community-associated cases were completely missed in North America. And in Australia, we also decided to license cephalospore for animal use in 2000s. And guess what? In around 10 years later, in 2010s, we were also seeing an increased number of CDI cases. Now let's talk a little bit about the emergence and spread of rabbit type 027. Canada was the first country to report such a significant increase in CDI cases that was linked to rabbit type 027. In between 1991 to 2003, their case number increased by 4 times. And among the elderly population, it increased by more than 8 times. And both the complication and mortality rates more than double. And that was because of 2 different lineages of rabbit type 027 that are resistant to fluoroquinolones. And if you have a look at this map over here, it shows that the 2 lineages basically arrived somewhere in North America, and then they were traveling back and forth between North America and Europe and UK quite a few times before they spread to other parts of the world. So how important is C. difficile? Well, it's ranked as one of the top 5 urgent AMR threats by the CDC in the US. It causes upwards of 500,000 infections and 29,000 deaths per year in the US, and that's roughly around 6% mortality rate. And in Australia, that equates to roughly around 11 deaths per week. It causes the healthcare system a lot of money, mostly because of increased hospital stay and treatment costs. Now, community-associated CDI cases is actually on the rise. In the US, 40% of the CDI cases are community-associated, and in Europe, that's 15%. In Australia, 30% of the cases are from the community. This is a study by David Eyre, who's in the UK. So he sequenced 900 plus isolates from human CDI cases and performed whole genome sequencing followed by SNP analysis. And his study shows that 45% of those CDI cases were actually genetically distinct from all previous cases. It shows that there's quite a significant proportion of the CDI cases over there are not acquired from another symptomatic patients, which totally refute the traditional notions that CDI is a hospital-associated infection acquired from another case. And this study shows that there must be another source causing all these C. diff infections outside of the hospital environment. Now let's look at what ribotype is circulating around Australia. The top ribotype in Australia is always ribotype 014/020. That's this one over here, and in red, and can be found in all states. And then the second most common ribotype is 002, followed by 056. Now that one is actually quite consistent. They're always on the top 5. Sometimes there are new strain that will pop in every now and then. For example, ribotype 012. That one is quite new. We think it might be originated from Asia. And then ribotype 017 is also very prevalent in Asia as well. So that may be where it's coming in from. So now let's have a look at what's going on in Asia. So this is a study performed by Deirdre Collins. She's a postdoc in our research groups, and this study was funded by Otsuka, which is a pharmaceutical company based in Japan. So they wanted to make vaccines for C. difficile, And before they do that, they wanted to know what strain is circulating around Asia, which is why they commissioned us to perform this study. So in Asia, the most common ribotype is actually ribotype 017, but it's ribotype 018 that's the most common in South Korea and Japan. So this study also included some strain from Indonesia, but we only have 7 from Indonesia. So if you guys want to give us some sample, feel free to contact me. So now let's just do a little bit of summary about what we just talked about, which is C. diff in humans. So CDI is a significant burden on human health. So we already established that. And then the C. diff colonization and disease is significantly driven by the use of antimicrobial. And then we also talk about the emergence of hypervirulent strains such as ribotype 027 and the increasing number of CDI cases. And we also mentioned a study from UK, which shows that there's a significant proportions of CDI cases that are genetically distinct from all previous CDI cases, and this challenged the traditional notions that CDI is a hospital-associated infection. Okay, so next we're going to move on to talk about C. diff in animal. So C. difficile has been isolated from all of these animals, and this covers pets, domesticated animals, wild animals, farm animals, and reptiles as well. So now I'm going to talk a little bit on some of the study that we did in Australia. So this study took place in abattoirs and farms all around Australia, and we collected 371 sheep and lamb sample. And then the finding was that we found C. diff in 6.5% of the lambs, but only 0.6% of the sheep. Now the lamb is a lot younger than the sheep, and that's why the prevalence is a lot higher in the younger animal. But this study took place before we know that the C. diff is mostly highly prevalent in neonatal animals, which is around the age of less than 7, at most 14 days. And in this study, the most prevalent ribotype is ribotype 101. Now, this is another nationwide surveillance study of C. difficile, and this time it took place in neonatal pigs. So this study included 21 farms all around Australia, and then we collected 229 fecal sample, and this time from piglets under the age of 14 days. And the prevalence of C. difficile is a lot higher than the previous studies. We found C. diff in 67% of the sample, and then the most prevalent ribotype is ribotype 014, which is the #1 ribotype in causing human CDI in Australia and as well as in overseas as well. But in Western Australia, the most dominant strain is actually ribotype 237. So a few years later, we decided to do a vaccine study for pigs. And because CDI is a toxin-mediated disease, the toxin itself are the prime target for vaccine development. So in this study, what we did was we inactivated the toxins and the cell surface antigens of C. difficile ribotype 014 and ribotype 237, because those two ribotypes are the most common ones in Australian pigs. So basically, we had 2 vaccine candidates from those 2 ribotypes, and then we were trying to see whether or not we can induce an immune response in a cell that would lead to protections for their piglets against C. difficile in a real commercial farm setting. So this study took place in a piggery farm owned by Australian Pork, and then we have Apriums, which is a veterinary service provider company that was helping us on the farm regarding vaccinating the pigs, setting up our study, collecting samples for us. And then the key findings is that ribotype 014 vaccine can confer cross immunity against the toxin from ribotype 237, but the opposite cannot be said for the ribotype 237 vaccines. And we also found that the piglets from the vaccinated cell have a much lower rate of C. diff colonizations compared to piglets from unvaccinated cell, which is a good thing. But the level of antibodies in the colostrum milk of both the vaccinated groups was not significantly higher than the control group. So this is something that we still need to work on, and it's on our to-do list. We just need to find the money to do it. So this is another one of our study, and this one is about C. diff in cattle and calves. And it's a collaboration with Meat and Livestock Association. They have a lot of farms all around Australia as well. And this study involved 25 abattoirs. 975 samples were collected from adult cattle and calves under the age of 7 days. And then the finding was the prevalence was the highest in calves under 7 days old, which is 56%. And then by the time they reached the age of 2 to 6 months old, it dropped to 4%. And then in adult cattle, the prevalence of C. difficile dropped further to 2%. And then the most common rabbit types in this study are rabbit type 127, 033, 056, and 126, all of which has been found in humans before. So how do we tell whether or not there's any interspecies transmission happening between animals and humans? There's actually quite a few different ways. You can do MLST, pulse field, or rabbit typing. See whether or not they belong to the same typing groups. That's one way. But obviously, some of the methods will have a better resolution than the others. You can also do MLVA typing. But currently, the gold standard is to do whole genome sequencing followed by core genome SNP analysis, and that stands for single nucleotide variant or single nucleotide polymorphism typing. So basically what it does is even if there's a single nucleotide that has mutated in the core genome, this method will detect it. That's why it's the gold standard for analyzing disease transmissions. And on the right hand side, those are the papers that use these methods. And before I talk a little bit more detail about those findings, I'm going to talk a little bit about the molecular clock of Clostridium difficile. So basically, a bunch of really smart people have come together and then they did a lot of experiments using whole genome sequencing, sampling, followed by mathematical algorithm calculations, and then they figure out that the mutation rate of C. difficile is actually 1 to 2 SNP per genome per year. And because of that, if two strain has 2 or less than 2 SNP differences in their core genome, they are considered as clonal, which means that transmission between the two sources are likely to happen. So this is a study from the Netherlands, and they were looking at rabbit type 078, which is also a hypervirulent strain. It causes severe diseases, it produces all 3 toxins, it's frequently found in the animals over there in Europe, and it's also associated with causing CA/CDI in younger, healthier patients. Actually, not patients, people in the community with no exposure to the hospital setting. So in this study, they collected samples from pigs, pig farmers, and clinical isolates from the hospital, and then they performed whole genome sequencing followed by SNP analysis. And they found there were 3 instances of potential interspecies transmissions highlighted by those 3 arrows over here, and those were clonal transmissions. And they have 0 SNP differences between them, and those strains were actually from pigs and the farmers. So it shows that the transmission were either happening between the 2 species, or it could be also because of the exposure to the common sources, which is the farm. So a couple of years later, the same research group basically built on what they previously found in the previous papers, but this time around, instead of focusing on what was happening in the Netherlands, they included rabbit type 078 strands from 22 different countries. Mostly from Europe and North America, there were a few strains from Asia and Australia. So the key findings were that there were clonal and highly related classes of rabbit type 078 from humans and animals. And there were limited geographical clusterings. In one of the clonal group number 1, it actually included a pig strain all the way in Canada that's clonally linked to the one that were causing infections in human CDI cases in UK. So that's intercontinental transmission across an ocean. And it showed that the spread of rabbit type 078 clones between farm animals and humans can actually travel across long distance. So this is actually one of our own studies. So we decided to sequence rabbit type 014 because it's the most common cause of human CDI in Australia. It's also the most commonly found in pigs in Australia. So in this study, we included 24 clinical isolates and 16 isolates from pigs. After performing whole genome sequencing and SNP analysis, we found out that there were 6 distinct clonal groups, 2 of which contain a mixture of humans and pigs isolates, and that's showing interspecies transmissions. And those two clonal groups are highlighted by the arrows, and that's clonal group number 3 and number 2. So both of which included strain from different states. Clonal group number 3 included strain from New South Wales and South Australia, while clonal group 2 included strain from Queensland, Victoria, New South Wales, and South Australia. And then there were no evidence of epidemiological link. The animals and the human were never in contact with each other. And then there were no geographical clustering. Furthermore, over half of the CDI cases in those two groups have no prior exposure to healthcare systems. Overall, it showed that there must be a persistent point source community reservoir that can travel across long distance, causing all this long distance interspecies transmissions. And at the time, we were thinking that it might be through food that contaminated food in the national food chain. So in summary, Clostridium difficile is an important pathogen and commensal in a wide variety of animal species. High prevalence of C. difficile has also been found in Australian neonatal animals, with many of the isolated ribotypes being clinically important in causing human CDI cases. We also provided evidence of national and intercontinental transmission of C. difficile between animals and humans. Okay, so now we're going to move on to talk about C. diff in the environment. So C. difficile can be found in diverse sources in the community. It has been found in hospital, household environment, in the nature, in seafood, meat, vegetable, and salad. And that's because of agricultural practices in recycling animals and human waste. So in piggery farms, for example, when one of the pigs is sick, the whole shed of pigs will get a shot of antibiotics. And because of intensive farming, they produce a lot of waste. And those waste will get treated in these kind of treatment ponds. But the thing is, these kind of treatment ponds is actually not effective in reducing the number of spores. And after it's being treated, the wastewater will get recycled onto farmland as irrigation water, or being reused as a washing water in the piggery farms, while all the solid waste gets trucked down to nearby farm to use as fertilizer to grow crops and vegetables. So in Western Australia, the treated wastewater from the sewage plant is actually being pumped out into the oceans far away from our shorelines, while a small proportion of it is also being used to irrigate local parks and replenish the groundwater. Meanwhile, the biosolids will eventually get recycled onto land, but it's important to know that C. difficile has been shown to survive the composting process. So we recently did a study with Water Corporation to look for C. difficile in wastewater treatment plants. Because the paper is not published yet, and we do have a non-disclosure agreement signed with Water Corporation, so I can't tell you the details of our finding yet. But it's actually quite comparable to what other people found in other countries. So in general, overall, C. difficile can survive wastewater treatment plants. And a high prevalence of C. difficile has been found in effluents, biosolids, and as well as the rivers that's receiving the treated wastewater, and that's in Canada. And there has been extensive release of toxigenic C. difficile strains into the environment, and then the most prevalent ribotype in this study are ribotype 078, 014/020, as well as non-toxigenic 010. Next, let's look at the receiving bodies of those treated effluents, manure, and biosolid. So these are the two studies I did when I was doing my PhD. And these are all, both of these took place in Western Australia. So we looked for C. diff in the public lawns and retail vegetables. So in total, 311 lawn samples were collected around the Perth metro regions, and 59% of those samples has C. difficile. And the most prevalent ribotypes is again ribotype 014 and non-toxigenic 010. Well, in the vegetable studies, I collected sample from 31 retail stores and farmer markets all around Western Australia. In total, I processed 300 root vegetables, and the key findings were the overall prevalence was around 30%, but potato have a prevalence of roughly around 50 to 56. Now, it has a range because I sampled both organic and non-organic potatoes, while the rest of the root vegetables are all organic veggie. And in the vegetable studies, the most prevalent ribotype is also ribotype 014. So these are the studies that were performed by our PhD students, and this is about the gardens around hospitals and residential homes. So Cecilia collected 159 samples all around the 4 major hospitals in Western Australia, and she found 60% of those samples were contaminated with Clostridium difficile. And again, the most prevalent ribotype in those samples are ribotype 014 and non-toxigenic strain 010. Now, when it comes to the sample collected from the residential homes, the manure have a highest prevalence, 83%, followed by soil around the garden, 79%, followed by compost. If the household like to do gardening, they will have compost around the house, that's 67%, while the bottom of the shoes have a prevalence of 32% for C. difficile. And again, the most prevalent ribotype is 014 and 020, sorry, 014 and 010. So this study is about C. diff in composted products, and when it comes to retail compost, 23% of them are actually positive for Clostridium difficile. And again, the most prevalent ribotype are the common ones that we see in humans when it comes to ribotype 014, 020, as well as ribotype 056. And ribotypes 010 seem to be very common in environmental sample, and it's non-toxigenic, which means we rarely see it in human CDI cases because it doesn't cause diseases. And we decided to sequence ribotype 056, and these are the strain on the right hand side. As you can see, we have 21 from humans, 4 from vegetables, 2 from compost, 1 from a vet clinic, and 1 from lawns. And after doing whole genome sequencing and SNP analysis, we identified 3 distinct clonal groups, 2 of which contain a mixture of humans and vegetable/compost isolates, and those are the 2 clonal groups highlighted by the arrows, and that's clonal group number 2 and number 3. So number 2 includes 2 human CDI cases and 1 from potato, and clonal group number 3 included 2 CDI cases from humans, 2 from compost, and 1 from potato. All these strains are from Western Australia, so this is local transmission, and the conclusion is this suggests environmental transmission, or at least a shared common source. So this is another one of our studies that look for C. diff in lake, ponds, river, estuary, as well as seawater. So in terms of prevalence, the highest one is found in lake and ponds, followed by river, estuary, while the seawater tested negative for C. difficile. Now in this study, the most prevalent ribotype is again ribotype 014. Ribotype 014 seems to be everywhere in Australia, and because it's one of the most it is the most common ribotype in causing human CDI in Australia. That's why we decided to sequence it. And in this study, we sequenced 26 isolates from humans, 7 from this study, which is from water. And after performing SNP analysis, we detected 7 closely related groups. Now this is not clonal, so they are more than 2 SNP difference in the core genome, but less than 10. So that's what we call closely related clonal groups. And 5 of those groups contain a mixture of humans and water isolates, and it shows that they at least have a shared common ancestor in the last 5 years. And overall, 19% of the CDI cases that were sequenced in this study share a recent genomic history with 1 or more water strains. So again, environmental transmission seems to be playing a role in CDI transmission. C. diff in environment is a relatively new field. There's only a handful of studies out there that look for C. diff in environments, which is why when it comes to comparing the genomes of environmental strain with human strains, there's even less paper out there. There's these are all the papers that do the genomic comparisons. 1 uses MLST, 1 uses MLVA, while 4 uses core genome SNP typing, and 3 of those are my papers. And I already talked about the ones about C. diff in water, followed by the C. diff in compost. So now I'm going to talk about this paper over here. Now, because there's a lot of genomic evidence out there already that say that there's a significant proportion of CDI cases that were genetically distinct from all previous cases. So those cases are not hospital-associated CDI. They are not acquired from another symptomatic patient. So it's very likely there's another community sources out there where people are getting infected or colonized. And second of all, we do know that there are a lot of diverse sources and reservoir of C. difficile in the community. And because of that, we started to wonder whether or not it's possible for C. difficile to be imported into the hospital either through soil compost, shoes, and once it's get imported into the hospital, does it cause CDI? So that's the focus of this paper. We're trying to answer that questions. To answer that questions, we did another study at Charlie's in 2019. So this study was funded by the Department of Health. It's a 2-year study that sort of got stretched to 3 years. So in 2019, I basically did a year-long environmental sampling all around the hospital. So I collected 300 floor sample in the main corridor of the hospital, and I also approached hospital staff, patient, visitor, so I can get a sample of the bottom of their shoes. I also collected 300 of those shoe sample, and then the prevalence of C. difficile on floors is 30%, while on shoes is 31%, so it's quite similar. But the thing is, toxigenic strains, it's very rare. The floor sample only have 0.3% of toxigenic strains, while the shoe sample only have 4.3% of toxigenic strains. And again, in this study, non-toxigenic ribotype 010 predominated both sample type. Ribotype 014/020, I did found a few of them, 9 in total, and all of which were from the shoes. And in this hospital in the year 2019, ribotype 014/020 causes 33% of the CDI cases, and because of that, we decided to sequence both of these ribotype, both ribotype 010 and ribotype 014/020. So over here in total, we sequenced 46 C. difficile ribotype 014/020 isolates, all of which were from Charlie Hospital in the year 2019. 27 were from CDI cases, 9 were from the shoe sample, and 10 were diarrhea sample that didn't meet the testing criteria to look for C. difficile. So we did whole genome sequencing, SNP analysis, and then this graph on the left-hand side over here is a pairwise comparison of our SNP analysis. So basically, you can have a look over here. It shows that majority of the strain, they are very different, like genetically very diverse from one another. There's only 2 strains that's clonal. That's 1 SNP difference from one another, and then we later found out that they were actually isolated from the same patient sample 4 months apart. So overall, there was no patient-to-patient transmission happening with ribotype 014/020, which is quite surprising to us because it's the most common cause of CDI in Australia. But there was no patient-to-patient transmission happening whatsoever in this study. And there was also no contaminated shoes to patient transmission that we can find. So, diverse genetic variations seem to suggest that the cases were likely originated from sources outside of the healthcare system. So, in the next step, we decided to include all our previous environmental samples. So, in total, we sequenced 142 ribotype 014/020, 65 of which were from humans, 17 from animals, 60 from the environment. So if you have a look at the legend on the left-hand side, it shows the sources of the isolates, and that's also indicated by the first column over here, while the gray colors in the second columns, it shows that the dark gray means that they are clonal, and then the light gray means they are closely related with 9 or less than 9 SNP differences in their core genomes. So after performing SNP analysis, we've identified 10 distinct clonal groups, 3 of which contain a mixture of humans, animals, and environmental strains. So that shows interspecies and environmental transmission. And those are the 3 clonal groups, 2 of which contain strains from different states. So that's long distance disseminations of C. difficile, while clonal group 3 are all were from Western Australia. We also have identified 16 closely related groups with less than 9 SNP differences in their core genomes, 13 of which contain humans, animals, and environmental strains. So, in conclusions, we provide evidence for frequent bidirectional spread between humans, animals, as well as the environment. While ribotype 010 is non-toxigenic and it doesn't cause any diseases in humans, but because it's so prevalent in our environment, we wanted to sequence it because we wanted to find out whether or not it's possible for environmental C. difficile to be imported into the hospital. So for this study, we sequenced 66 ribotype 010, and if you have a look on the legend on the left-hand side, it will show the sources and then the number of strains that belong to each group. So the human floor shoes and then isolates from the CF clinic and a hospital garden, they were all from the hospital environments, while the compost, lawn, vegetables, water were from the wider community. And after performing whole genome sequencing and SNP analysis, we identified 6 clonal groups, 5 of which only cluster the floor and the shoe strain together. And that showed that there's a recent transmission event that happened at St. Charles Gardner Hospital. And that could be from the floor to the shoes or from the shoes to the floor. We can't tell the directions from the SNP analysis. Furthermore, the floor and the shoe strain were very closely related to the strain from compost, lawn, hospital gardens, as well as the water isolates. And that showed that they have a recent shared ancestry in at least the past 5 years, and then transmission from one source to another is very likely. And again, it showed possible importations of ribotype 010 from the community into the hospital. Now, even though this is not such a big deal in Australia because it's non-toxigenic, it doesn't pose any risk to human health, but because C. difficile in environment is such a new field, we don't know much about what's happening in other countries. So if a country have a lot of toxigenic strain in the environment, it is possible for those toxigenic strain to be imported into the hospital through the environment, for example, soil, compost, shoes, floor. So the transmission of Clostridium difficile seems to be a lot more complicated than what we originally thought a decade ago. It's not just a hospital-associated infections, it's actually a One Health issues. So the waste product of humans get treated in the wastewater treatment plants, and then the biosolid get composted, which C. difficile can survive, and that get used as fertilizer to grow crops and vegetables, so that contaminate our environments, and the humans can come into contact with the contaminated environments or the contaminated food. At the same time, C. difficile is very prevalent in the production animals, and the production animals produce a lot of waste that some of them don't get composted. They can just apply directly onto land, while some get composted before they get applied to land. And then there are studies in the US and Canada and Europe that shows that the meat are also contaminated with C. difficile. Prevalence a little bit low depending on where you are, but in Australia that's not the case. At least so far we haven't found any C. diff in any of our retail meat. Anyway, C. difficile on food can also be spread to humans, so it's basically a cycle, and C. difficile is being transmitted between humans, animals, and the environment. Summary. So there are diverse sources of C. difficile in the community. C. difficile can survive wastewater treatment plants and composting. Recycling of animals and human waste is a significant risk for zoonotic and environmental transfer of C. difficile and community acquisitions of CDI. In Western Australia, C. difficile has been found on public lawn, vegetable, compost, garden, river, pond, lakes, and estuary. And genetically indistinguishable or closely related strains of C. difficile has been recovered from humans, animals, and the environment. So overall, CDI seems to have a zoonotic or environmental etiology. So what can we do about this problem then? So we can start putting cephalosporins into production animal for a start. We can also improve the treatments and management of animals and human waste. We can increase surveillance of C. difficile in diverse sources, not just focusing on the hospital. And we also need better communications across different sectors, for example, farmers, clinicians, veterinarians, and policymakers. Conclusions: C. difficile from humans, animals, and the environment share a recent common ancestor, and there are local, national, and intercontinental transmission of C. difficile clones. Point source of C. difficile in the community is likely due to agricultural recycling of animals and human waste product. There's also compelling evidence that CDI may have a zoonotic, foodborne, or environmental origins. So One Health in a genomic framework is an optimal approach to both understanding and reducing the transmission of CDI. So lastly, I'm going to talk about some of the projects that we are currently working on so that you guys can have a better understanding of what we are doing as a whole research groups. So currently I'm working on the NHMRC Idea Grant study. So that's a 4-year study and it was funded for $1.28 million. It's a collaboration with UQ. So I'll be looking at all the human CDI data over the past 15 years and collating all the typing data that will ever perform in Australia. Then that includes the one done for humans, animals, and all the environmental studies. And then we'll also be doing perspective studies, so that's looking at C. diff in clinical cases, farm animals, gardening supplies, vegetables, households, environment, as well as hospital bathroom of high-risk wards in 2 different hospitals. One's Charlie in Western Australia, and another is Royal Brisbane Women's Hospital in Brisbane. And so far, currently, all of our environmental studies are WA based. So with this study, this NHMRC Idea Grant Perspective study, we'll start to get some interstate environmental samples. So I'm fairly excited about that. So once that's done, we'll also be interviewing key stakeholders about the use of antimicrobial and their waste management protocol in animal farms, as well as horticultural practices. And then we're gonna follow and find out the fate of the animals and human waste after they treat it. And then basically we'll collect all this information and data, and all those information will be used to construct a system dynamic model. Don't ask me what is a system dynamic model. I have no idea. So that part is going to be performed by our collaborator at UQ. So basically it's a model that's about the transmission of C. difficile, and then they are going to use the model to do a cost-effective analysis to look for a potential interventions that's the cheapest but have the most impact economically and health-wise, and that's going to be used to inform policy, practice, and government guidelines. And another study that I'm currently working on is a GP study. So because C. difficile in the community, it's underreported, because in hospital, if a patient have 3 or more episodes of diarrhea in one day, their stool sample will get tested for C. difficile. But in the community, the GP won't ask for a C. difficile test unless the patient has been having diarrhea for 3 weeks. And because of that, CA-CDI is underreported in Australia. So in this study, we'll be working with 2 networks of GP, one in rural and one in metropolitans. And then the GP is going to be the one recruiting the patients, and then they're going to ask the patient to send a sample of the stool sample to Puffwest. And then we have a very good working relationship with Puffwest. So we basically just gonna go there and get the stool sample after they finish testing it for the presence of toxin genes and toxins using EIA. So anyway, after we gotten the sample, we'll find out whether or not they have C. difficile and what ribotype it is, because Puffwest don't do typing. They only want to know whether or not they have the toxins in their stool sample, while we are interested in the typing and the strains and the whole genome sequencing and all that. So in this study, we'll find out the true incidence of CA-CDI. We'll also perform case control study to look at the risk factors, And then we'll also go to the do some household sampling of the patient to find out whether or not their household is contaminated by the same strain. And then another study that I'm doing is looking at C. diff in children with cancer, and that one is a collaboration with some pediatric oncologists and infectious diseases specialists at the Perth Children's Hospital, which is just across the road from Charlie Hospital. And there's also a reptile study that is a master student project at UWA. It's a one-year study, so that should be ending at the end of the year. And then we also have studies about C. diff in cat, dog, horses, pet foods, and those are Murdoch vet students. So we do get a lot of vet students coming in from Murdoch because they are doing their master degree, and that's 2 years, but they are required to do a research project that's gonna last at least 3 months over the course of 2 years. So yes, we do have students coming from all different kind of university. So next, we are also doing like wastewater treatment study. Like I said, I was collaborating with Water Corps, and then the paper is close to ready for submissions. And then I'm planning on taking the collaboration to the next step by helping them identify which treatment system is more effective at reducing C. difficile spores. And that one I'm planning on using culture and real-time PCR. So that's a wastewater treatment study. And then we also have a CRC study. So this one we just found out that it got funded a couple of days ago, and that one is a 10-year study. So CRC stands for Cooperative Research Center. So it's a it involves a lot of different sectors and a lot of different university, including industry partners. So everyone basically put in a little bit of money, and then once the project get funded by the government will put more money so that it became a bigger pot of money for us to use and do our research. And I think the next step is for people to actually meet up and to discuss how they'll divvy up the money for whatever research they want to do. Anyway, the focus of this project is to look at AMR in wastewater, agricultural, business, food, and the environment. Other than that, we're also doing the C. diff, the vaccine in pig study that I mentioned. And then Roche also have a study that they are funding us to do. So that one is a rapid real-time PCR machine that they want to have in the emergency department. So it's going to be a point of care detection for pathogens. So that machine can detect the pathogens in under 20 minutes. So they want to test whether like how good it is. And then we are also doing cystic fibrosis study. So looking at C. diff in CF patients, we're also studying a biobank for respiratory sample. And then Dan, who's our bioinformatics persons in our research groups, he'll also be doing the genomic analysis on Pseudomonas. And he's also interested in the evolutions and genomic of C. difficile. And this project involves a citizen project where basically the citizens who live in a remote part of Australia collect the soil sample and then they send it to us, and then we'll do our stuff. We also have study that took place in Thailand, Cambodia, Vietnam. So these are different PhD projects. So the one in Thailand is already finished. So the student already graduated. He's currently back in Thailand. He's an MD who wanted to do a PhD so that he can become a lecturer in a university in Thailand. So he's a double doctorate. And then these two, Cambodia and Vietnam, is still ongoing. And then we can also do phenotypic and genotypic characterization of strains. So that's a lot of different assays looking at the sporulation, germinations, tissue culture, those kind of things. And this one is bioinformatic based. Okay, so lastly, I would just like to acknowledge everyone in my research group. So this is mostly everybody in our research group in this photo. And we are a multicultural and multi-institute research group. We have people who are employed by UWA, Murdoch, ECU, and Curtin. So that's the 4 universities in Western Australia. And I would also like to acknowledge all our collaborator and funder. So we previously have studies that was funded by the Department of Health, and then we also have study that was funded by the NHMRC and the ARC. So NHMRC only fund human research, and ARC fund everything else, including research that involve animals. And because C. difficile it impacts both humans and animals, we can double dip and apply for both. Some researcher can do that. So, and then we also previously got the money from Otsuka company to do the C. diff in Asia study. And then Roche always asks us to do a little bit of study for them every now and then. And then Gamma Healthcare, this is a company that sells a lot of disinfectants and cleaning product directly to hospitals. So they previously also funded a study that I didn't mention here. And then Pathwest, we work very closely with them, and these are all the diagnostic laboratory, both private and public in different states of Australia that we work with. And then there's Australian Pork, Meat and Livestock Association, APM Animal Health, so they were helping us with the national animal studies. HGLF do our sequencing for us, and then we use Policy for their supercomputing services, Water Corporation for the wastewater study. Rich Grow is a company that make compost and a lot of gardening product. And Monash University will do all our hamster model. All our hamster model that need to take place, Monash University do it for us. And then our latest collaborator is from UQ. So they are the modeler who's going to be doing the modeling and doing the cost analysis for us. And that's the end of my talk. Any questions?
Okay, thank you so much, Sue Chen, for the very extensive and elaborate talk. You guide us through all the aspects of One Health in regards to your research in Clostridium difficile. You mentioned about how Clostridium difficile in regards to human health, also in animal health, environment, and also the solutions and current projects. So we really did get an overall and complete understanding of your work. So I'd like to open this Q&A session, if you don't mind. Yeah, okay. So we have a question from Dr. Dian. So she's our colleague here in the Faculty of Medicine, Mataram University. If you don't mind, I'll read the question in the chat box here. So her question is, can you elaborate more on how vaccinating animal against C. difficile could benefit human and environmental health? So that's the first question. And the second question is, she was wondering in regards to what policy, either hospital or national policy, was developed as the result of the study that showed that the RTO14 is not transmitted from patient to patient, but rather from the environment. So yeah, you can answer the question, Sichan. Thank you.
I'm sorry, can you repeat the first question again?
Okay, it's in the chat box, but I'll repeat it for you. First, Dr. Dian wanted to know more on how vaccinating animal against C. difficile could benefit human and environmental health. So I guess we'll go from one question to the other. Okay.
So when you vaccinate the animals, hopefully the animals will have less C. diff in their guts, and because there's less colonizations in the animals, there will be less C. diff contaminations in their waste products. So eventually, over time, that will lead to less contaminations in the environment, and that will lead to less exposure to humans, and that will lead to less CDI cases in humans.
Okay, so for the first question, perhaps Dr. Dian, do you have anything that you'd like to continue, or is there any aspects for the first question that you would like to know more?
Thank you, Dr. Dewi. My next question is how often that you need to vaccinate the animal? Is the vaccinations like influenza, in which you need to repeat it every year, or once vaccination for the whole life of the livestock is enough? That's the next question. Sorry, I'm not a microbiologist, so my question might be very basic. Yeah.
Okay,
And
so
yeah.
go on.
Do you want to answer that first, perhaps, Suchan?
Okay, sure. I'll answer one question at a time because otherwise I have memory issues, people. So we haven't really done that study yet, but when we were planning the vaccine study, we were planning on giving like the vaccine to the sow twice a year right before they about to give birth. So we were planning on looking at that and see whether or not they needed a booster shot and how often they would need it. Like, do they need it once every single year? And because in pigs, they give birth twice a year. We were, that's one of the things we need to look at, but I'm guessing you probably need to have a booster shot once a year. But we haven't done that study yet.
Is there any evidence that support that support the notion that community that live in area that vaccinate their livestock to Clostridium difficile is is benefited by the vaccinations or less having less infections of Clostridium difficile? Is there any empirical evidence on that?
There's currently no evidence of that because there's no vaccines available against C. difficile for animals. There's actually not even one for humans. There is one that's under development by Pfizer that's currently in phase 3. So hopefully we'll get a vaccine for humans soon. But for the animals one, there's not much progress at all.
Oh, I see. Okay, so that's in your presentation it is under research. Is it correct? So it's not applied yet to the industry, to the livestock industry.
No, it's not applicable to the industry yet. We're still in the early phase of developing the vaccines, and that study that we did was quite preliminary. So we were planning on expanding it by increasing the number of cells and then doing like more study to extend the finding, but we were having difficulties finding the funding to do that study because even though Aprium Animal Health, so that's the company that's provide the vet services to a lot of farm in Australia, they were very keen on funding the study, but at the same time, they don't want to fund the entire study. They were hoping to fund part of the money and then write up a research proposal and apply for government funding that's going to match their contributions. So we're still in that phase trying to get the study funded, but right now I'm slightly distracted by all the other studies I'm doing, but it's on a to-do list. We'll get there eventually.
Okay, thank you.
Thank you. And there's another question also from Dr. Dian. The second question in regards to the policy, if I'm not mistaken. Dr. Dian asked about whether the policy, either in hospital or national level, was developed as the result of the study that showed that the RT014 was not transmitted from patient to patient, but rather from the environment. So how the follow-up of that research?
Well, trying to change the policy is always so difficult. You need to present the policymaker with a lot of evidence to back up your studies, and making changes in the hospital is a lot easier because we're already like working with those people in the healthcare sectors, and that's very easy to apply. But because ribotype 014 seems to be transmitting predominantly in the community. Now, in the community, things get very complicated. There's a lot of different factors that's in play. We still trying to narrow down like where are the sources is being transmitted. There's a lot of potential sources that can play a role. They may all be playing a role. So if you want to control everything, for example, lower the number of C. difficile contaminations in lawn, compost, and vegetables, for example, you need to be working with the compost making company. You need to liaise with the horticultural sectors. You also need to talk to the people that are actually recycling the wastewater for both humans and for the animals. Now in Australia, there's no guidelines. So for the animal waste can just be applied straight to land. We do have some rules for the humans waste product, but the rules are not based on the quantification study of spore-forming pathogens. They were only looking at E. coli and coliforms. So those die very easily, but spore is very hardy. So C. diff can survive through the entire wastewater treatment system of the human sewage pipelines. So we need to talk to a lot of people and then convince them to make a change. In order for us to do that, we need to provide them with a lot of evidence that can't be moved and have to be very convincing.
Okay, thank you, Su-Chan. Dr. Dian, do you have a follow-up questions in regards to Su-Chan's answer?
No, I'm satisfied with the answer, Dr. Dewi. Thank you.
Okay, thank you so much for the question, Dr. Dian. We greatly appreciate it. So the next question is from, hold on, Ida Kade. Okay, and her question is, so this student wanted to know, is there any evidence of C. difficile in seawater? And she noticed that there's no prevalence in seawater, and why is that? I mean, is it, so how is it like C. difficile in seawater? Is there any evidence that it could be a source or not? And if not, then why would it be the case?
So in my study, I sampled quite a lot of seawater sample up and down the western coast of Western Australia. So I think it was close to like 90 something sample of seawater, and they were all negative for C. difficile. Now I was a little bit surprised by that finding, especially given the fact that I know Water Corporation has been pumping out the treated wastewater that has C. difficile into the oceans. But the reason why we didn't find any is probably because it was so diluted, and they pump it out far away from the shores, like more than 1 to 4 kilometers away from the shore, and they don't dump it into the ocean in one go. That it's basically get diluted because the pipe has a lot of holes. So the treated effluents will slowly get diluted by the seawater, and because of that, it doesn't really reach the shorelines where the people are swimming around.
Oh, okay. So if you don't mind, I'll translate.
Another thing, sorry, can I just follow up on another thing?
Yeah, sure.
So that's what's happening in Western Australia, but in Brisbane, they are actually using the treated wastewater to grow crops. So they're using it as an irrigation water for growing vegetables. Now, I'm pretty sure over there they do advanced wastewater treatments before they do that. But again, they were only looking at Salmonella and E. coli when they were doing the safety test. Nobody was looking at any spore-forming pathogen.
Okay.
So I'll be interested to find out whether or not the irrigation water have C. diff.
Okay. So if you don't mind, I'll translate it for the student. Jadi dari yang disampaikan oleh Dr. Su-Chan bahwa terkait dengan identifikasi daripada Clostridium difficile pada air laut sampai sejauh ini masih inkonklusif. Jadi salah satu dugaan yang disampaikan oleh Dr. Suchen adalah bahwa ada kemungkinan dari limbah yang diduga mengandung si difficile itu begitu sampai di laut kan terdilusi sehingga bisa jadi tidak cukup untuk kemudian bisa diidentifikasi. Jadi bahwa ada atau tidak sampai kemudian air laut itu terkontaminasi sampai saat ini masih belum bisa dipastikan. Kemudian yang kedua adalah bahwa pada saat ada satu penelitian atau bukan penelitian ya, di Queensland itu air limbah itu dapat digunakan kembali, tentunya setelah mengalami suatu proses. Nah, hanya saja di sana itu untuk melihat ada kontaminan bakteri itu masih terfokus pada beberapa patogen, dan itu mengeksklusikan atau belum mengidentifikasikan patogen-patogen yang kemungkinan berbasis spora. Jadi kan kalau Clostridium difficile ini kan dia ada tahapan sporanya ya. Nah, itu belum diidentifikasi. Jadi sampai saat ini belum bisa disimpulkan apakah kemudian ada atau tidaknya. Kalau sementara ini dikatakan tidak, itu lebih karena mungkin ada faktor-faktor lain yang menghambat atau belum berhasil untuk diidentifikasi. Jadi masih menjadi area untuk diteliti. Gimana, Ida? Apakah ada yang mau di follow up dari pertanyaan ini?
Tidak, Dok. Cukup segitu saja.
Ya, makasih.
Terima kasih, Bu Dokter.
Oke, sami-sami. Oke, are there any other question? Atau silahkan mungkin ada yang ingin bertanya, boleh tanya langsung pakai bahasa Indonesia atau bahasa Inggris, nanti kita bantu terjemahkan. Boleh juga langsung di chat box. Nah, mumpung ada Dr. Suchen di sini yang dengan senang hati menjawab pertanyaan. Ada lagi mungkin, atau ada yang punya ide penelitian atau terbersit ada penelitian tertentu? Okay, while we're waiting for other questions, I have several things that I also would like to confirm. Okay, my first question is that it seems like there's a disparity in regards to identification of Clostridium difficile between the developing and developed country. Although you did mention that even in the developed country, it's still underdiagnosed, but at least research there is much more prominent than it is in the developed world. And I think one of that is that in regards to diagnosis and several research method is for identification is through full genome sequence and PCR, which may not always be available in low resource countries. So my question is that would there be a more, how would I say it, a more a diagnosis or ways of identification for Clostridium difficile that would be more approachable and would be feasible to be conducted in developing world?
For identifying C. difficile or identifying whether or not the patient has CDI or both?
Both, in human and also in animal.
Okay, so to diagnose CDI, you don't really need to do, because if you do PCR based, it's a lot faster, but there's actually other method that's actually equally fast, even or maybe even a bit faster as well. So if you use EIA, so EIA basically it's just a very small kit. You just grab a little bit of poop sample and then you just stick it straight in, and then a little bit of time, add the reagents, and then basically it'll just let you know whether or not the stool sample contain any toxin A and toxin B. So because even if you do have the even if the stool sample are positive for the toxin producing genes of C. difficile, that doesn't necessarily mean that the toxin is actually being produced and causing diseases, right? So right now in Pathwise, what they're doing is after the stool samples became tested positive for the toxin producing genes, they still do the EIA, which is actually looking for the toxin itself. So that kit is actually quite cheap. I'm not quite sure how much off the top of my mind, but that one is not labor intensive, very quick, very simple, and it doesn't take up much space, unlike some of the big machines that Pathways is using. So that would be a good option. And then another option is to do culture. So with culture, it's a little bit more time consuming because you need at least 2 to 4 days. But the benefit is you actually get a sample of the bacteria itself. So if you want to do follow-up studies like find out the AMR profile of the bugs, you can. Unlike EIA, which is basically just a diagnosis whether or not there's toxins in the stew. So those two methods, it's quite doable in a developing country.
Okay, interesting. Thank you, Suchan, for the answer. Are there any other participants that would like to ask a question for Suchan? Ada yang mau bertanya lagi? Okay. Okay, in that case, I'll try to wrap up the presentation for today. So it seems like tackling Clostridium difficile requires while it is known that tackling a Clostridium difficile requires a comprehensive approach and collaboration between animal health, human health, environmentalists, and also because it has a very diverse source. And there is a disparity of identification of the understanding of Clostridium difficile in the developed and developing country, hence remains an underdiagnosed, particularly in the developing countries. Identifying the ribotype in developing countries is also an area to seek, as understanding this may lead to research for development of rapid diagnosis and also vaccine development in human and in animal. And this would be a interesting research area for collaboration between students in medicine and also in other fields, like in veterinary medicine and also animal science. And for the last part, I'd also like to point out to the students that are here in Indonesia is that, jadi adik-adik sebetulnya ini saya lihat ada beberapa mahasiswa dari latar belakang atau program studi yang berbeda. Dan dari apa yang disampaikan oleh Suchan bahwa untuk penanganan si difficile ini kan memerlukan pendekatan yang multidisiplin. Dan ditambah lagi sebetulnya masalah ini itu underdiagnosed baik pada hewan pada hewan ternak maupun pada manusia. Ini bisa menjadi area kolaborasi penelitian yang menarik untuk adik-adik, baik yang kemudian bisa didiskusikan dengan dosennya atau dengan pembimbing atau dengan pakar yang ada di bidang keilmuan adik-adik, bahwa masih belum banyak diketahui terkait dengan identifikasi pada Clostridium difficile pada hewan, pada manusia, dan juga pada lingkungan. Jadi gimana kondisinya dari Clostridium difficile yang ada di Lombok atau di Nusa Tenggara, ditambah dengan sebetulnya kan di Lombok dan juga di Sumbawa atau NTB itu kan adalah area yang sangat kaya dengan hewan ternak gitu. Jadi kira-kira apakah ada kekhasan daripada Clostridium difficile yang ada di Lombok dengan di negara lain gitu. Jadi ini merupakan suatu area yang masih sangat sedikit ya diteliti dan menarik kemudian untuk dikaji. And you also mentioned before that in regards to identification of Clostridium difficile, there's not many research in Southeast Asia. Am I correct, including in Indonesia?
There are a little bit of research about C. diff in Asia, but it's mostly from Japan, Korea, Taiwan, and Hong Kong. Okay. And not much is happening in Malaysia and Indonesia.
Yeah.
And I think like the CDI cases in Asia might be a little bit lower compared to what's going on in Australia and Europe and North America as well, because C. diff can actually be separated into 5 different clades. And the one that's predominant in Asia are clade 4. Now, clade 4, a lot of them are non-toxigenic. And because it's non-toxigenic, it doesn't produce diseases. And maybe because of that, it also make it harder for the toxigenic strain to come in and set up shop, because it's basically competing with them as well. So that's one of the hypotheses that we think why C. difficile infection is not that common in Asia, but in countries such as Hong Kong and Taiwan and Japan, where there's a lot of international travel, is actually pretty common. Like they do get occasionally the ribotype 027, like the hypervirulent strain, and sometimes they do get outbreak as well. Oh, and then the ribotype 017 that's very prevalent in Asia. It's also causing a lot of outbreaks in Europe. So I'm not quite sure whether or not the same is happening in Asia, because nobody has looked at it. And the reason why ribotype 017 is causing outbreak in Europe is because it's very resistant to a lot of different drugs.
Okay. Thank you, Su-Chan, for the explanation. So that's quite interesting to actually explore and monitor. And that's great news that the toxigenic type is not prevalent, and we hope that it continues that way. Okay, so I guess that would be the end of our Q&A session. And thank you so much, Su-Chan, for your time. It was really an honor for us to have you here. And I'll give it back to the MC for the next session.
Thank you very much, Dr. Dewi. Ladies and gentlemen, the organizing committee would like to give an appreciation to our guest speaker, Su-Chan Lim, PhD. A certificate will be given by Dr. Dewi Suryani, Master of Infectious Diseases. To Dr. Dewi, time is yours.
Okay, so thank you, Su-Chan, for appreciation that you have given your time to us and provide valuable information and shared your work. We'd like to present you with this certificate as our speaker for today in our first guest lecture in 2022. And we really hope that it doesn't stop here, but we can continue with other works and also future talks. So this is initiation phase, I guess, for future work in the future. Yep. So maybe you'd like to have a word or two?
Well, I'm hoping the COVID might die down a little bit, because right now we're having a lot of COVID in Western Australia because our borders just opened like 2 months ago. And I'm hoping to go to Lombok because you guys are so close to Bali.
Exactly.
Maybe in a few months I'll be able to see you in person.
Okay, that's great. Because it's so close. You used to have a direct flight from Perth to Lombok, so we hope to meet you, and perhaps we could extend to other collaborations too. I'm sure that the faculty would be more than happy if there are future collaboration that we could initiate.
Yes, our research group is always interested in collaborations. Like, definitely reach out to me if you guys are interested.
Excellent, thank you so much, Su-Chan. Oh, we actually have our dean here. I was wondering, Dr. Hamsu, apakah ada dua patah kata yang mungkin mau disampaikan? Maaf, Dok, saya baru lihat. We have our dean here, Pak Dekan. Oh, hold on. Oh, hi, Dr. Hamsu, how are you?
Yeah, hi, Dr. Dewi. Hi, Su-Chan Lim.
Hi.
How are you?
I'm good. Thanks for inviting me to give a speech about C. diff.
Okay. Yeah, thank you very much for your sharing. And I'm sorry I couldn't join at the beginning of this session. But anyway,
okay.
I think your lecture is very interesting. Yeah. Okay. Thank you, Dr. Dewi.
Yep. Thank you, Dr. Hamsu, for your time and support for this event. Okay. And yeah, so I'll pass it on to the MC. Silahkan Di Ayah.
Thank you, Doctor. Once again, thank you very much, Suchan Lim, PhD, for the very educative and interesting lecture. And thank you very much, Dr. Dewi Surani, as today's moderator. Our next agenda is a post-test, which will be conducted for 5 minutes. The post-test link will be sent in the chat box, and participants will be redirected to our website where the post-test will be done. We wish all participants to fill in this post-test. Thank you. Time is up. Thank you very much to all participants participants for completing the post-test. So ladies and gentlemen, today we have several door prizes which will be given to participants with the best pre-test score, best post-test score, the participant with best question during today's lecture, and also to one lucky participant who fills in our feedback after the event. Lucky recipients will be announced after the event on our website www.fk.unram.ac.id. Before we close our event, we will have a photo session first. So to all participants, we encourage you to turn on your cameras, please. Thank you. To organizing committee, time is yours.